As we prepare to study EEG biofeedback, sometimes referred to as neurofeedback, it's important to remember that it too, like all biofeedback training, is based on operant conditioning. While classical conditioning forms an association between two stimuli, operant conditioning forms an association between a behavior and its consequences. Let's look again at the organization of the nervous system. General biofeedback involves operant conditioning of the peripheral nervous system, particularly the autonomic nervous system. EEG biofeedback involves operant conditioning of the central nervous system, particularly the brain. The German psychiatrist Hans Berger was the first person to prove the existence of electric potentials or voltage fluctuations in the human brain by using an amplifying machine. He recorded the first human EEG, or electrocardiograph. His research work centered on intracranial blood circulation and blood temperature. Berger studied the work of Richard Catton, a Liverpool surgeon who was studying action potentials in animals. In 1924, he made the first EEG recording in a human. We can think of the brain as a structure, an organ. It has both chemical and electrical properties. But the brain is also where humans derive their sense of consciousness. While the brain is both physical and biological, consciousness is neither physical nor biological. The cerebrum or cortex is the largest part of the human brain, associated with higher brain function such as thought and action. The cerebral cortex is divided into 4 sections, called lobes: the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. The EEG signal arises from the cerebral cortex. This is a thin layer of nerve cells that is divided into 6 thinner layers, which are striated according to the neuronal cell type, cell density, interconnections with the other cortical cells, and connections to other areas of the brain. The pyramidal cells in the outermost layer, where the tip or apex of the nerve cell dendrites exist, are believed to be the source of the EEG voltage fluctuations. The activity of the excitatory and inhibitory synaptic potentials in these dendritic trees produce the voltage wave detected on the scalp.